Hi Year 11, this is a review of the Biology B2 March mock exam that we've sat just recently. What's going to happen, we're going to go through the mark scheme for this paper question by question, and what you need to do is make sure that you are doing improvements in green pen to any questions that you got wrong, also looking for any missing marks that you somehow missed on the paper. Okay, so we'll get right into it. We've got question 1. It says figure 1 shows 4 cells A, B, C and D. Each cell is surrounded by oxygen molecules, and it says that oxygen molecules move into cells or out of cells. So it's talking straight away here about the movement of molecules in and out of cells. Anything to do with movement of molecules or in or out of cells is to do with diffusion. So that leads us quite nicely into the first question. It says name the process by which oxygen moves into or out of cells. That is diffusion for 1 mark, folks. Okay, so you need to put diffusion for that one. Question 2, well 1A part 2, into which cell A, B, C or D will oxygen move the fastest? So notice this is just for 1 mark. Answer is A. And the reason for this is because if we look closely at cell A, the rules for diffusion are that molecules or particles will move from an area of high concentration to an area of low concentration. So by high concentration, that means that there is a lot of those particles actually within a certain area. So you can see here on the outside of the cell, there's loads and loads more particles, so a higher concentration than there is inside the cell, because there's only a few of them inside the cell there. So if we look at the question, it's asking which cell will oxygen move the fastest, so into which cell, and it's going to be cell A for that reason, because there's a higher concentration on the outside. If you need to make any corrections off this page, now is your time to do it, folks, because we're going to move on in a second. Okay, so we're on the next page now. This question is 1A part 4. Why do cells need oxygen? You could have said, well, the main thing that you could say for this answer is that the cells need oxygen for respiration. So respiration was okay for a mark. Or you could have also said to allow the cells to release energy. So that was a nice straightforward 1 mark question. And then we've got a 6 mark question that generally for my groups was answered quite poorly. We'll look at that now. So it says in this question you'll be assessed on using good English, organising information clearly and using specialist terms where appropriate. So it is a quality of written communication question, and it does expect you to use scientific language throughout it. So it says the cells in large plants and animals are organised into tissues, organs and organ systems. Describe each level of organisation of cells and give examples of these in plants and animals. So it's basically asking you in the question to describe what a tissue is, what an organ is, and what an organ system is, and to give an example of each of those for a plant or an animal. Okay, so we'll go through a number of things that you could have said now. So you should now be able to see a copy of the mark scheme for this 6 mark question on your screen. Just a final thing before we go through it though, it says specifically do not refer to the parts of cells. So that means you cannot refer to cell organelles like the nucleus or mitochondria or ribosomes or anything like that. You're just talking about the different levels of organisation. Okay, so let's look at the mark scheme. So it says specifically here then, for between 1 to 2 marks, there is a description of a level of organisation or an example of a level. So by that it means you give a description of a tissue, a description of an organ or an organ system, or you've provided an example of one of those levels. For 3 to 4 marks, there's a description and an example, or a description of levels or examples. And then finally, for 5 to 6 marks, there's a description of levels of organisation with some linked examples. So it has to include all levels of organisation, so that means tissue, organ and organ system, and it needs to have at least 2 linked examples in there, preferably 3. Okay, so the actual biology points that you could have made for this question then, in order to get your actual 6 marks. First one, it describes a tissue. So tissues are made of groups of similar cells working together. That was your description of a tissue. And your example, in animals, you could say muscle tissue, epithelium tissue, or glandular tissue, or epidermis. So those are your tissues in animals that you could have mentioned. And then in plants, we've got the mesophyll, the xylem, and the phloem. Okay, so we've got the spongy mesophyll, which is in the leaf, which is where palisade cells surround for photosynthesis. We've got the xylem for water transport, and then we've got the phloem for nutrient transport. The next level of organisation, we have got organs, and it specifically needs a description of organs to say organs are made of groups of different tissues that are working together. So you could say that in the heart you've got muscle tissue and you've got epithelial tissue. Okay, so it's a description of two different tissues working together. Some examples. This was one that students mostly did really well on, so they gave loads of good examples of organs. The stomach, the heart, lungs, kidneys, those are all good examples of organs in animals. If you were giving organ examples in a plant, and the stem, the root, or the leaf in plants, those were some other good examples that we've seen as well. Okay, finally, description of some organ systems is that it's a system made of groups of different organs that perform a specific function. So it's different organs working together to do a bigger job inside the body. So you could have said an example might have been the digestive system, you could have said the respiratory system, you could have said the circulatory system, or the central nervous system. Any of those would have been okay. You could have also mentioned that the flower was an organ system in the plant. Okay, finally, you could have also said that an organism is made of groups of organ systems working together. So if you think about humans, humans have a working digestive system, a working nervous system, a working circulatory system. It goes on, there's more to it than that as well. You could have given any example of a named animal or plant. So there's plenty of potential here to get 6 marks. Unfortunately, a lot of students either didn't read the question correctly or they had not revised the basic descriptions of those different levels of cellular organisation. So that is an area that needs to be improved. If you need to add any corrections to your paper for this question, which a lot of you will need to do, pause the video now and get down some of these points in green pen, please. Okay, question 2. This is quite a nice one to get stuck into. So it says green plants use light energy to make their own food in the process of photosynthesis. Complete the word equation for photosynthesis. So it's talking about plants and photosynthesis in this question. So we've got carbon dioxide plus water, and very basically it's just a basic knowledge and understanding question. They react to give us glucose and oxygen. So that there was a really nice 2 mark that you could have easily got some marks on. Next question, a little bit more tricky. It's getting you to apply your knowledge instead. So it says one product of photosynthesis can be used by plants to make other useful substances. Complete table 3 to name these substances. So we're thinking of something now that is used to strengthen cell walls, and the answer for that, the named substance, is cellulose. Next one, something used for storage. You could have said starch, fat, oil, or lipids. So basically anything that is fat. So fats are really good for storing energy. And then we've got the final one, something that is made using nitrate ions. There's a number of examples that we could have said for this one. Some of the main ones we'll just list off now. Protein, amino acids, enzymes, chlorophyll, any named hormone in the plant. So auxin was one of the more popular ones that got named. You could have said DNA or nucleic acids, which are the components that make up DNA. This question was also one that was generally answered quite poorly, so if you do need to make any corrections, do that now please, or you can do it at the end of this page when we move up to there. We'll go to the next question, which is question 2A part 3. Name one tissue used to transport substances around the plant. You could have either said the xylem or the phloem for this question. So the xylem is used to transport water, and then the phloem is used to transport nutrients around the plant. Okay, so nice, quite straightforward question. Okay, so we'll read the part for question 2B, and then we'll move on to the next page so we can continue it there. So it says marigold plants can be bought from garden centres in the spring. The owner of a garden centre needs the plants to grow as quickly as possible. Scientists investigated the effect of change in the light intensity and the temperature in the greenhouses where marigolds are grown. Okay, so we've got figure 6 here, which is showing the relationship between the light intensity That's measured in a unit called lux, and the time until the plants are ready for sale in days. Okay, so the lower the number of days, the faster the plant is growing. That's what you've got to think here. So that was sort of something that a lot of people missed out on. They thought if it was a higher number it would grow faster. It's not what the graph is showing us. So make sure, please, that you analyse and pick out what the graph is actually showing, because that was an area that got a lot of people confused. Okay, so our first question then says the rate of photosynthesis is affected by limiting factors. What is meant by limiting factors? You need to give here some idea of something that is affecting the rate of photosynthesis. So it's something that is limiting the rate of photosynthesis, stopping it from increasing. So you could have said something that prevents photosynthesis from increasing, or something that stops the rate of photosynthesis getting faster. So anything like that is okay. So some examples of these things that can be limiting factors. We've got our basic ones, which are temperature. We've got the water content, light intensity, which is what this investigation was looking at. But also we've got carbon dioxide level as well, because carbon dioxide is one of the things that are needed for photosynthesis to take place. Okay, so we'll move down and look at the next question. Okay, so it says now, what was the lowest light intensity to produce the fastest growth rate of the marigold plants at 25 degrees Celsius? Use information from figure 6. Okay, so let's go and look at the graph and look at how we could answer this one. So our key bits from the question that we've got to try and pick out first is that it is only for the plants at 25 degrees Celsius, and you're trying to find the lowest light intensity to produce the fastest growth rate. So let's move up. Okay, I'll make this a little bit zoomed in. Look at it in a bit more detail. Okay, so it is only this line here that we are interested in. The other line is for 15 degrees, so do not pay any attention to this line here that's being pointed out now. It's only this one that we want. And we're looking for the lowest light intensity where we've got the maximum rate of photosynthesis, so the quickest time that it grows. If we look at here, this is showing the time until the plants are ready for sale in days. So the lower the number of days, the faster it is growing. And you can see, if we point out on the graph, that the point where it reaches its lowest point is about here. I apologise if it's not 100% accurate, but I am doing this with my finger on the iPad. And you can see there that the light intensity is around about 80,000 lux. Okay, so it's 80,000 lux was your answer. However, the examiners will accept if you said anywhere between 80 to 82,000. Okay, so it could be 80 to 82. And that was your answer for that one. So it's all about actually looking at the graph and figuring out what it's actually showing you first, before you just jump straight into it and pick out a number. Okay, so we'll go on to the next page. Okay, so question 2b, part 3. Suggest why the owners of the garden centre might decide not to use 25 degrees Celsius and the light intensity you gave in part B2 when growing marigolds for sale in greenhouses. So you're thinking now, if they've got to maintain that temperature of 25 degrees Celsius, then it's going to cost a lot more money than if they had it at room temperature instead. So your answer for this one, folks, was because it may have a higher cost, or it may not be cost effective. And if we look at the next question, the data on the graph was collected at a carbon dioxide concentration of 0.04%. On figure 6, draw a line to show what you would expect to happen if the marigolds were grown at 25 degrees Celsius and a carbon dioxide concentration of 0.4%. Justify where you have drawn your line. Well, I'm going to give you the justification first, and then we'll go through where it actually should be. So because there is more carbon dioxide that's actually available for the plant, because if we look at the numbers, 0.4% is a higher concentration than 0.04%. Therefore, if there's more carbon dioxide, then there is, it's going to increase the rate of photosynthesis. Okay, so you've got 1 mark for stating that there will be a faster rate of photosynthesis, and for the second mark you could say that this is because there will now be more glucose available, so that glucose can be used for growth. You could also say for your second mark that carbon dioxide is no longer a limiting factor, or you could say that the line plateaus, it gets flat, because other factors are now limiting it instead. Okay, so we'll look, I'll just put those on the question sheet for you so you can see them. If you need to make any corrections for this point so far, then do that now please, because we're going to go on to looking at the graph and where you should have drawn your line now. Okay, so it was 1 mark for saying that there's going to be more carbon dioxide, so there's a faster rate of photosynthesis, and your second mark for your explanation why that is. So more glucose available, CO2 is no longer a limiting factor. Okay. Okay, so let's take a look at the graph. We'll just get that zoomed in now, so get rid of this. And bring the graph into screen. Okay, so because we've now got a higher amount of carbon dioxide, that means that it's going to increase the rate of photosynthesis, therefore the plants are going to grow a lot faster and they're going to be ready for sale in a shorter period of time. So what we were expecting you to do was to show that the rate of photosynthesis has increased, and you did that by drawing a line that's at a steeper gradient, because it's growing a lot faster, but starts to curve off at a point anywhere below 25 degrees Celsius. So if you did this, that would also be accepted, or if you did this, it would also be accepted. Okay, but it had to be below the 25 degrees Celsius mark. Okay, so that was showing that you understood how to plot that onto a graph. Okay. Okay, we're now on question 3. This is looking at a quadrat. So it says, for 7 days, students investigated changes in the population of yeast cells in a flask that contains sugar solution. The students used a counting chamber to estimate the number of yeast cells in the sugar solution each day, and a counting chamber has a grid where 1 millimeters equals 1 millimeter and has a depth of 0.01 millimeters. So it's giving you the dimensions of the counting chamber there. And it says 4 days after the start of the investigation, a sample was put into the counting chamber. It says figure 7 shows the view of the counting chamber through a microscope. So this is what they would see if they were looking down through a microscope. And you can see that we've got the yeast cells distributed throughout the counting chamber. Okay. So it says the students decided to include in the count all the yeast cells partly inside the upper and left sides of the grid, and not count all the yeast cells partly inside the lower and right sides of the grid. So what that actually means is, if we look at this counting grid in a bit more detail, they would include the ones that were partly in on this side and at the top, but they would not include the ones that were partly in at the bottom and on the right hand side. Okay, so that was the student's method, and that comes into play a little bit later on in the question because you need that. Okay, so it says, for now, suggest a different method of overcoming the problem of yeast cells partly inside the grid. So we had to give some different solution to the one that the students had already decided on, and you could have said any number of things for this. Some of the more popular ones, and the ones that are on the mark scheme, were to count all of the yeast cells that were more than half inside the grid, or ignore the ones that were less than half inside the grid. So you could have also said count all of the cells along the edge and divide it by 2. Any other reasonable methods as well, where you are counting the number of cells around the outside and giving some sort of method to get a more reliable representation of those. So I'll put the one on the mark scheme in the question box now for you folks. Okay, so moving on to the next page, it's a continuation of this question. So we've got 3A part 2. It says a number of yeast cells per millimetre cubed of solution can be found using the formula. Number equals the actual number counted divided by the volume of liquid in millimetres cubed. So it says using the students, notice that it is in bold, using the students method, not another method, calculate the number of yeast cells per millimetre cubed in the solution and show clearly how you work out your answer. So I am going to talk you through exactly how to do this. It was really straightforward, so we'll get the diagram back up. Okay, so we can now see the diagram that shows the distribution of yeast cells, and the student's method was to count the ones that were on the left hand side that were partly in and the ones at the top that were partly in. So these areas here, so from there, they would include those, but they would ignore the ones that were on the bottom and on the right hand side. So let's count them then. So we've got 1, 2, 3, 4, 5, 6. We're not including the one next to this one because that one's touching a line. 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28. We are not including this one here, this one here, or this one here, because the student's method only states that we should be counting the ones along the left that are partly in and the ones along the top that are partly in. Okay, so that gives us 28 in total. So we now have to use that number of 28 with our equation that we're going to bring back in now. Okay, so we'll move that off the screen and put that back there. Okay, so we know our actual number counted was 28, and the volume of liquids, we can calculate that by multiplying all of the edges of the counting chamber. So that was 1 times 1 times 0.01, which gives us a total of 0.01. Okay, so the calculation that you then have to do, just get rid of this, is 28 divided by 0.01, which should give you an answer of 2,800. Now some people managed to get error carried forward marks here. The mark scheme says to allow 1 mark for clear evidence that they've counted 28, 1 mark for clear evidence of 0.01 millimeters cubed. However, if you've got a different count, you could get up to 2 marks if you did the rest of the calculations correctly. Okay, so out of 3 marks there, quite a nice easy one, and a lot of people just didn't follow the student's method. A lot of people were just counting them, counting all of the squares that were there. You've got to read what the question's asking you to do, folks. Okay, so 3B, Table 4 shows the results collected by the students. Describe the pattern of increase in the population of yeast cells between day 0 and day 3. This one was answered really, really well. You need to say that the population increases rapidly, and a lot of people noticed that the pattern was that it multiplied by 4 times as many each day. So it was 1 mark saying that it increased, and then your second mark saying that it increased by 4 times each day. So like I said, that one was answered generally quite well. Most people tended to spot the pattern. They could have also got a mark if they said it just increased by a higher number after each day. So you didn't have to spot that it was by 4 times as many each day. For 3B part 2, it says suggest 2 possible reasons for the rapid decrease in the population of yeast cells after day 5. So we can see here after day 5 it's at 11,200, and then it drops quite drastically down to 2,100 instead. Okay, so points that you could have said for this then, it was 2 marks, 1 for each point. You could have said a lack of food, sugar or nutrients for 1 mark, a lack of oxygen. You could have said that there was an accumulation of waste or toxic waste. Or you could have also said anything to do with predation. So you could have mentioned that the yeast were being eaten by something. Okay. The mark scheme says though specifically to ignore death. It will not accept death as an answer, but it would have accepted that the pH of the solution had changed. Okay, so we'll move on to the next question. So this question here is looking at enzymes. So it's got question 4A. Protease enzymes digest proteins. What type of substance is produced when a protein is digested? Just a basic knowledge and understanding question. Your answer for that, folks, is amino acids. So whenever we have proteins that get digested, they form amino acids. Okay. So the next part of the question for 4b, it describes an investigation into the digestion of protein by protease. So it says egg white is a protein. Boiling changes egg white from a clear liquid to a white solid. The student did the following, so it goes through the different steps of the experiments. They put 10 centimeters cubed of liquid egg white into a measuring cylinder, put the measuring cylinder in boiling water until the egg white went solid, put 5 centimeters cubed of protease solution into a test tube, put both in the measuring cylinder and the test tube into a water bath at 15 degrees Celsius for 10 minutes. So they're not actually in contact with each other at this point, because it shows you the diagram here for step 4. They're just in the same water bath at 15 degrees Celsius. Step 5, they added the protease to the solid egg white. So this protease solution would have been added to the egg white. And then step 6, they recorded the volume of solid egg white after 24 hours. So they could see how much solid egg white there was left. And then we've got the liquid egg white here that's been broken down by the protease enzyme. Okay, so the actual question then for this, we'll look at that now and see what it's asking us to think about. So bring it over. So it says, in step 4, why were the solid egg white and protease solution put into the water bath for 10 minutes before being added together? So the reason for that, if you're putting two substances in a water bath, the point of that is to get them at the same temperature. So the answer was to get them at the same temperature. Part 2, it says to calculate the volume of solid egg white digested after 24 hours at 15 degrees Celsius. Use figure 1 to work out your answer. So we'll look at figure 1. Just move this out of the way, make it a little bit smaller. And we've got figure 1. So this shows, it might be a little bit pixelated, but you can see it clearly enough. This shows that at the start of the investigation, there was 10 centimeters cubed of egg white. We can see that there on the measuring cylinder. And then after 24 hours, it had dropped down to this point here. Oops, sorry, that's not come out properly. It's down to about here, which is around about 7 on the measuring cylinder. So to calculate how much of the egg white has been digested, we do 10, so that's our starting amount, minus 7, which gives us 3 centimeters cubed. Okay, so that is the amount that has been digested. Okay, and that was a question that for some reason or another a lot of people were making simple mistakes on. So it wasn't really that difficult. Okay, so we should have got 10 minus 7, giving us 3 centimeters cubed. I'll just put that on the paper for you so you can see it. Okay, so we've got 3 centimeters cubed for that one. We'll move on. Let's group these together. Okay, should be good now. Okay, so if we move on, 4C part 1, describe the relationship between temperature and the volume of egg white digested at temperatures from 10 degrees Celsius to 40. So we are only looking specifically from this point here at 10 down to 40 here. Okay, and we can see that every time the temperature goes up by 10, so from 10 to 20, the volume doubles. So most people were able to spot that link there with the pattern. So from 20 to 30, it goes from 4 to 8. 30 to 40, it goes from 8 to 16. So you got 1 mark for saying that there was an increase. So as the temperature increased, so did the volume of egg white digested. And then your second mark was for mentioning that it doubles in number each time. So that was quite a nice, straightforward one there to get stuck into for that one. So not such a complicated set of data that you've got. Okay, we'll go over to the next question. Okay, so we've got, not this one here. So we've got 4C part 2. Student 1 wrote a conclusion. It says, protease works best at 40 degrees Celsius. This conclusion may not be accurate. Suggest how the investigation should be changed so that student 1 could find a more accurate value for the best temperature. And this is a 2-mark question, so you've got 2 available marks to get on this one. The way to get the first mark was by saying to repeat the investigation at a range of different temperatures. So that is not just doing it at 10 degrees Celsius intervals, it's doing it at temperatures between those. So you might say for your first mark, repeat the experiment at different temperatures. So that'd be okay for a mark. And then your second mark you would get for giving some suggestions. So repeat at 35, 40 and 45 degrees Celsius. So it's just giving some suggestions as to the numbers that you would use. So that was a 2 marker, that one was quite a nice one as well. Okay, moving down to 4C part 3. It says, student 2 wrote a different conclusion. It says at 50 degrees Celsius, protease is killed by the high temperature. Why is the conclusion written by student 2 not correct? Well, basically, protease is an enzyme and it's not a living organism, so it cannot be killed. So you could get a mark just for saying that the protease is an enzyme and cannot be killed. So that was 1 mark for that. It was quite nice and straightforward. Way in which student 2 could correct the conclusion is by saying, instead of using the word killed, he could mention that the enzyme would be denatured. And what that basically means is the active site of the enzyme, so the place where the substrate locks onto, that is going to change shape so it can no longer catalyse the reaction. Okay, so it's all about using the key language in that question. Okay, next page. Okay, so we've got a respiration question. It says this question is about respiration. Anaerobic respiration is different from aerobic. The equation shows anaerobic respiration, so we've got glucose, which is breaking down, it's the incomplete breakdown of glucose to give lactic acid. Aerobic respiration does not produce lactic acid. Give 2 more differences between anaerobic and aerobic respiration. Well, there's a number that you could say for this. You could say that aerobic respiration requires oxygen. You could say that. You could also say that aerobic respiration produces carbon dioxide. Or you could also say that it produces water. Those are all differences between each kind of respiration. So I'll just make them a little bit clearer for you there. The mark scheme also allowed it if you said that anaerobic respiration releases less energy than aerobic respiration. That's because in anaerobic respiration it's the incomplete breakdown of glucose. You could also say that anaerobic respiration causes fatigue in the muscles. And that's about it, folks, for the points that you could say. So just to repeat that, so we've got anaerobic respiration requires oxygen, so this bit here. It produces carbon dioxide and water. It releases less energy, so anaerobic releases less energy. And it also, anaerobic causes muscle fatigue. Okay, so it was quite a nice one there. Okay, so we'll move down to the next one. Blood contains glucose. The amount of glucose in the blood at the start of a long distance race will not be enough for a runner during the race. The runner does not eat or drink during the race. How do the runner's muscles get the extra glucose needed to run the long distance race? So this question was not answered very well. A lot of people hadn't revised this topic, it seemed. So this question was all to do with a chemical called glycogen. Okay, and glycogen is stored in the muscles. And essentially what happens to our glucose when we've got too much glucose inside our body, it stores some of that excess glucose as glycogen in the muscles. So glucose is stored as glycogen in the muscles. So that if we ever get to a position where we've not got enough glucose in our blood, we can then convert that glycogen back into glucose. So glycogen in the muscles is converted back into glucose. And that's really important for events such as marathon races where you've got plenty of glycogen stored in your muscle, so you've got a large supply of backup glucose there for when you need it. Okay, I also accepted a couple of other points to this one. You could have also said that fat could be converted to glucose. That was okay for a mark. If you've mentioned anything about a hormone called glucagon, you could mention that glucagon converts glycogen back into glucose as well. Other than that, there weren't really many other points that you could say to get any extra marks. So it was those three there that we've just mentioned. So we've got glycogen being stored in the muscles, glucose is stored as glycogen there, and then the glycogen can be converted back into glucose throughout the race, so that the long distance runner can use that glycogen for respiration. Okay, that's about it for this question. We'll move on to the next one. Okay, so we've got 5b part 2. It says lactic acid builds up in the runner's muscles, causing an oxygen debt. What is meant by an oxygen debt? Well, what you need to say for the oxygen debt, a lot of you were saying that it's when we've not got enough oxygen, which I suppose technically is right. However, it wasn't in enough detail for the mark scheme. The mark scheme requires you to say it is the amount of oxygen required to oxidize or break down the lactic acid in muscles. Okay, so it was that key bit towards the end that a lot of people weren't saying. So it's the amount needed to oxidize or break down lactic acid. So it's getting rid of that toxic lactic acid that's bad for us and removing that from our muscles. Okay, and that was a 1 mark question for that. Okay, so if we go down and have a look at question 6. So this one's on cell division. It's on mitosis and meiosis. There's a complete table for you to compare mitosis and meiosis for one complete cell division in humans. So what I'm going to do, I'm going to go through this one in a bit more detail because this is one of the ones that, once again, a lot of people were losing easy marks on. So it was a 3 marker and lots of people were losing them. So the number of times a cell divides, we're going to go through mitosis first. So we'll just minimise this, put it up here, come back to that a bit later. So mitosis is the replication of normal body cells. So if we start with a normal body cell, inside that body cell it's got chromosomes, and in humans there's 23 pairs of chromosomes. Okay, so that gives us 46 in total, which links in quite nicely with one of the points in the question. If it's showing you data for an animal or something that's got a different amount, you just got to remember that you need to give your answer as either pairs or individual chromosomes. Don't just assume that the examiner will know what you mean. So what happens in mitosis is this body cell will split, divide once to give 2 identical copies. Okay, so it's dividing once, so it's 1 division, which makes 2 new cells. Okay, so that was it for mitosis. If you need to make any notes on that on your paper now, you can pause it. We're going to rub it off in a second, and we are going to go through meiosis instead. So I'll get rid of this. Okay, so this time for meiosis. And meiosis is a little bit more different. So we start with a here with a diploid cell. So that's something that's got a full amount of chromosomes. It's got these pairs of chromosomes. It divides once to make 2, and then again to make 4. The key point for this, though, folks, is that the number of chromosomes halves as we get further down. So at this stage here, the chromosome number halves, so at this point here. And what that basically means is, if we look back at the question, it just affects the last part. Okay, so just forwarded a little bit there. So for mitosis, number of times a cell divides, it's once. Makes 2 cells. We have 46 chromosomes. For meiosis, it divides twice. Makes 4 gamete cells. And that has half the number of chromosomes at 23. Okay, so that was 3 marks. Really, really easy that we should have been getting, but not enough of us were actually getting those, which was a little bit concerning. We need to make sure we're revising that little bit there on mitosis and meiosis, please. 6b, one type of colour blindness is caused by a mutation of a gene on an X chromosome. A colourblind male must have inherited colour blindness from his mother. Explain why. Well, the chromosome pattern for males is XY. So male is XY and female equals 2 X chromosomes. Therefore, if the gene is on the X chromosome and the male is colourblind, then that means that his X chromosome must have come from his mum. So if we set this out as a genetic cross, we might help to explain it a little bit clearer. So we'll do that now. So we'll make this a little bit smaller, and we can set up our genetic cross now. So we'll do a Punnett square. We'll do the mum on the top, and let's say that this X chromosome here has got the gene for colour blindness on it. We've then got the dad, who is XY, and we'll do a cross. We've got 2 X's. This one here is our guy in the question, so he's inherited this X chromosome from the mum. We've got the dad down here. The other options, we'll just finish this off so it looks clear. So we've got XY, XY. So it's this one here. So he's inherited the X chromosome from the mum. So we'll go through the mark scheme specifically for this then. So let's just make this a little bit bigger. So you've got 1 mark for saying that the male is XY. And the second is for saying that he gets the X chromosome from the mum. So that in red. Okay. So it's pretty straightforward as well, that one was. Okay, so that's it for this page. We'll move on. Okay, going on to the last question now then. So we've got a question here on fossils and how they're being formed. So it says, evidence for early forms of life comes from fossils. Fossils may be formed from footprints in mud when the mud changes into rock. Describe in detail 2 more ways in which fossils may be formed. Notice though it's a 4 marker, so it will require you to describe this in quite some detail. Okay, so some of the points then that you could have mentioned, we're going to discuss some of those. So the first thing that you could say for one of the descriptions of how fossils can be formed, you could talk about mineralization, which is 1 mark, and then your second mark you get for actually describing what that is. So you describe it as decay taking place and then minerals replacing some of the lost material. Another one of the points that you could mention is that fossils can form from hard parts of animals that do not decay. So fossils form from the parts of animals that decay. So by this what we mean is something like the bones or the teeth of an organism, so none of the soft tissue that can easily decay. Other things that you could say are that fossils can be formed when some of the conditions for decay are not present. So fossils form when okay. So by that what we mean is if there's no oxygen present, then that will mean that bacteria and other microorganisms can't decay the remains of the organism. So it's when there's no oxygen present, it's when it's a really dry environment, so there's a lack of moisture, and it's also in an environment where there is a really low temperature, so when fossils can be formed or preserved in ice. The other one that it would accept on the mark scheme was fossils being formed in amber as well. So if you think about the example of mosquitoes, think Jurassic Park, you've got mosquitoes being preserved in amber. Okay, those were some of the main ones that you could have mentioned for the marks on this, but it was a 4-marker. You needed the method by which the fossil could be formed and then a description. Okay, so moving on to question 7b. If we look at this one, we've got another graph question where we've got to look at some data. It says the approximate age of some fossils can be calculated using radioactive carbon dating. Carbon exists in non-radioactive and radioactive forms. After an organism dies, the proportion of radioactive carbon in a dead organism decreases at a constant rate. It takes 5,730 years for half the radioactive carbon to decay. This time is called the half-life of the radioactive carbon, and figure 6 shows how the percentage of radioactive carbon changes over time. So if we look at the graph, we can see that as we get to 50% of the carbon remaining, that is one half-life. Then if that halves again, down to about 25%, that's 2 half-lives. So every time the amount halves, that is a half-life. Okay. So for question 7b part 1, suggest why estimates of the age of fossils using this method will probably be more accurate if the fossil is recent rather than if the fossil is much older. So it's pretty obvious. I mean, once we get down to this point here, past 6 half-lives, it is really difficult to measure the percentage of carbon, or the radioactive carbon, that is remaining inside the fossil. So for the mark for this one, you literally just need to say there is a higher level of radioactivity in more recent fossils. So I'll just put that in here, so higher level of radioactivity in more recent fossils. What that basically means for the scientists is that it's easier to determine what percentage there is and therefore how old the fossil is. Okay, so that was literally all we needed to say for that one. If we go over to the next question. Okay, so question 7b part 2. Recently a human fossil was found. Analysis showed that the fossil had 28% of the original radioactive carbon remaining. Estimate how long ago the human died. Use the information provided in the question and figure 6. So what we're going to do is we'll bring figure 6 over here and then we'll look at the graph. Okay, so we've got the graph here now, and we are looking to find out how old a fossil with 28% of the original radioactive carbon is. So if we use the data in the graph, so we'll go up to about 28%. So if we look on the y-axis, this is showing the percentage of that original radioactive carbon remaining. We go up, so we've got 0, 10, 20, and each of these little squares represents 2. Therefore, the point where it's at 28, I'll try and mark it on here as accurately as I can, but it is just about there. So what you have to do is draw that line across until it meets this point here. Oh, sorry, not that one. I'm drawing the wrong one. That's not 28%. This is 28%. So about there. And then where that meets, draw down. So you can see, it's not very accurate on here, but you can see that that is roughly about 1.8 half-lives. Okay, so we know straight away that the fossil is 1.8 half-lives. So we'll put that down, so it's 1.8. And we know from looking at the previous question that a half-life, or the previous information, that a half-life is 5,730 years old. So it takes 5,730 years for one half-life to happen. So in order to calculate the actual age of the fossil, we do 1.8 multiplied by 5,730, and then that should give us a total of 10,314 years. Okay, so that was a nice 3-mark question there for that one. Okay, moving down. So this is the last one now. There are very few fossils of the earliest forms of life. Explain why. So basically what this question is asking you to think about is what life was like during the early stages of the Earth's development. So we know that life at the start of the Earth's development was soft-bodied. That basically means there was no bones, there was no hard tissue, there was no teeth, it was soft-bodied. So think of a jellyfish. Okay, so all life was soft-bodied. Therefore, if all the life was soft-bodied, then that meant that nothing could be preserved as a fossil because all of the soft tissue would be decayed. So there would be no fossils due to decay. So for that one, you got 1 mark for saying that life was soft-bodied, and your 2nd mark for mentioning that they would all decay. Okay, that is it. Can you make sure if you've got any questions, you come and see me after half term, please.